My friend and colleague, Dr. Shanna Swan, who received her PhD at Berkeley and then was Director of Reproductive Health in the Department of Environmental Health at California. And then I met her on a National Academy panel where she was asked to analyze data relating to male reproductive health and chemical exposure. And at first, it was very clear she was skeptical that there would be anything, and then in the late 90s published an important paper on relationship of sperm count decline in Europe and the United States to temporal trend over the 20th century. She's been working to integrate information from endocrinology and developmental biology and toxicology into her epidemiological research ever since. And she was actually here for a while where she published that men in Missouri, in Columbia, have about the lowest sperm count in the world. I think after that, she beat it out of town. And she's now Dean of Research and mentoring and professor at Mount Sinai in New York, and she'll talk to us about stress and environmental chemicals and reproductive health. Shanna Swan.
So this is going to be a different kind of talk. I hope it'll be enjoyable, and I appreciate the invitation. Really, it's wonderful to come back here. If I can advance the slide. There we go. So what I'm going to talk about is a bit of personal history and a little bit of history in the field, and I'm going to talk about plastics and phthalates, then fetal hormones in relation to a phenomena which Fred von Saal actually is the foremost author on, intrauterine position effect, and then fetal hormones in relation to phthalates and something that I've spent a lot of time on, a weird measurement called the anogenital distance. How many people have ever heard of anogenital distance? Not too bad, yeah. A lot of kids have, by the way. It's a lot of street talk about it. You can look it up in Urban Dictionary. Anogenital distance in animals and in humans. And then I'm going to throw in a new wrinkle, which is the stress of the mother and how that may interfere with anogenital distance and also might interfere with the relationship between AGD and the phthalate associations that I'm going to show you. And then a little bit about what I'd like to do next. Okay? All right, so that's our house. Not anymore. That was our house, 714 Ingleside. I had the pleasure of having dinner there 2 nights ago. It's owned now by David and Ann Meir. And as you can see, we were pretty happy to get this house and to be here when we came in 1998. At the time, I was conducting something called I was conducting the Study for Future Families to look at this question that Fred had talked about, semen quality. And we wanted to see whether semen quality was different in Missouri, in Minnesota, in San Francisco, New York. And we launched this Study for Future Families and recruited women between 1999 and 2002. Why women? Odd, right? Turned out to be a really good thing to do. But the reason we did it was because we wanted actually to get through the women to get the men. And it's very hard to just ask a random man to give you a semen sample. And you don't know what the population base is for the men who give you one. Maybe they're all infertile, or maybe they're all fertile, or maybe they have whatever axe to grind. So, but if you get partners of pregnant women, almost all pregnant women come into prenatal care. So you know who you're getting and who you're not getting. So that was the design of that study. And it was conducted in these 4 cities. And you see, bing, right there, right? Columbia, Missouri. And that turned out to be really, really lucky. It was actually suggested by our study section, or our project officer, that we pick a place in the middle of the country that might have some rural exposure, some pesticides, and we did. So what we found and this is just one slide on this because it's not what my talk is about, but I just wanted to give you this, since I brought you this far on this, is to show you that Missouri, which is the light color, has significantly lower sperm concentration, percent of motile sperm, total motile count, compared to the other centers. And there was the strongest contrast was between Missouri and Minnesota. So actually, men in Missouri had about half as many, 58%, as many moving sperm as men in Minneapolis. It was pretty dramatic. And it actually caused a lot of press, including this cartoon from the Columbia Tribune, pointing out that everyone was aware of this finding. But this actually has not been solved. If somebody wants an interesting problem to work on, we don't know if that's still the case, and we don't know why. Although we did get some findings on pesticides, which I don't have time to get into, but we're not clear about the pesticide story. But at that point, I was going to Japan to talk about this study, sitting next to a colleague from CDC, John Brock, and he turned to me and he said, "Shanna, you should study phthalates." I go, "What? How do you spell that?" That was my introduction to phthalates. And why? And he said, well, at CDC, we know now that there's a lot of them in people's bodies, in the bodies of pregnant women, and we know these are reproductive toxins because in animals, they cause a lot of problems to the male offspring. So I thought, I could do that. Remember the study for future families? Well, what I did in that study, just on a whim, after it was about 1 year in, I said, let's just get urine. I can't honestly tell you why I said that, but it was the smartest thing I ever did, because it turned out that this urine was golden, no pun intended. It allowed us to do the next study. So before I go into that, I want to tell you a little bit about these chemicals. Okay, we haven't really talked much about them today. So at the end of the Second World War, the petroleum industry was growing, and there was a lot of oil, and there were a lot of byproducts of the production of oil. And the production of phthalates utilizes the byproducts of oil production. So it was a natural fit, and it was sort of natural that at that time there would be a growing interest in plastics, and that's what happened. So next. So I don't know how many of you saw The Graduate, but Mr. McGuire is asking Dustin Hoffman, lecturing him and saying, I have one word for you for your future, and it's, who knows what the word is? Plastics. And so lots of people followed that advice, and plastics became everywhere in our homes and everywhere in our diet. And I love this ad, plastics, an important part of your healthy diet. Okay, and so this was it was the golden age of chemistry, better living through chemistry, and plastics were going to solve every problem we had, except maybe not quite all. So one of the components of plastic that makes it soft is a phthalate called diethylhexyl phthalate. Big mouthful, I'll call it DEHP. Okay? So DEHP is in many products in our home and in our hospitals. And what it is, it's in this tubing. It makes the tubing soft and flexible. And what's unfortunate is when the nutrition, the nutrients, and the air and so on are going through that tubing, actually the phthalate is not bound to the plastic, and it comes off of the plastic and gets into the media and gets into the baby. And this has been shown in many studies that babies have higher levels of this chemical in their body, particularly in the intensive care nursery. But it's also in our food. And in fact, DEHP, our primary source, if we're not in the intensive care nursery, is going to be in our food. And that's tricky because we can't look for it, we can't find it, we can't prevent it, we don't know where it is. So that's the chemical that I've been focusing on now for about 10 years. The plastics industry, the chemical industry says that don't be ridiculous. There's only a tiny amount of this chemical getting into your body. And so I want to give you a little brief picture into how important a tiny bit of a hormonally active chemical can be for the developing fetus. So what I have to tell you, and we'll see more about that later, is that diethylhexylphthalate has a very special property that it interferes with androgen production. It doesn't work through the androgen receptor, but it interferes with testicular production of androgen. So as a result, when the male is in utero and exposed to phthalates, he will get just a little less testosterone. Why should we worry about that? So I want to talk about the intrauterine position effect, because I think this is the best story to tell you why a little bit of hormone change matters. So here are some pups in a uterine horn, and you can see they're pretty close to each other, and this guy gets some hormones. He makes some. He gets them from his mother. He gets them from this neighbor. He gets them from this neighbor. Right, Fred? Fred spent a lot of time counting these. And here's a schematic of them. And this is the uterine horns, and the pink is the females, and the blue is the male. So this is a 2 male female that is between 2 males, and this is a 2F male between 2 females. And it turns out that that position makes a difference throughout life. This will induce changes, and the amount of hormone that they're getting from their neighbors is pretty small. So here is a nice study which relates the sex of the pup and fetal position to the estradiol level measured at birth. So this over here is the most female-female. So this is a female who has no male neighbors. And this is the most male-male. And that's a male with no female neighbors. And what you can see is that as you go from female-female, the most female to the most male, you get a decrease in estradiol. But what's really interesting to me is right here. There's no difference. This is a female, this is a male, they have the same estradiol. And if you go to testosterone, you see the same thing, even more dramatically. So you see this female-female with this very low level of testosterone and fourfold higher in the male-male. So what difference does this make? Why do we care about this? Well, it turns out that the sex of the pup's neighbor influences the pup's development. This is just an example of the females between 2 males. So this is a 2M female, okay? And that female pup is going to mount other females, is going to be less attractive to males, is going to be less fertile, and she is going to have a longer or more masculine anogenital distance. Now, I'm going to define this, but just to tell you right now that this is the distance, as the name says, from the anus to the genitals. It's a sexually dimorphic measure, and it's longer, much longer, maybe twice as long in males as females. So when this female gets this little shot of testosterone from her neighbors, she gets a more masculine anogenital distance. And it turns out that this distance really matters, and I'll show you why. How about humans? Well, much more limited data. We don't have big litters. It's harder to study this. But there have been some studies, and here's one that showed that if you look at females, males who have a female co-twin, so they're getting a little shot of estradiol, they are more female in their sensation seeking, they have more sexually dimorphic behaviors. It affects their sexually dimorphic behaviors, it affects their reproductive fitness. It actually affects the ratio of the 2nd to the 4th finger, which is another sexually dimorphic measure, and it affects their birth weight. This study did not look, unfortunately, at anogenital distance. Another study, which I love, is a Finnish study which looked at female twins. So female twins with a female co-twin or a male co-twin. This is a remarkable study because the Finns keep wonderful, wonderful records. And this person went to the church yard and got the records of all the births and the marriages back to 1734. Pretty amazing. And found the twins and then looked at two things. What was the probability that a female ever married if she was, she had a female co-twin versus a male co-twin, her chances of marrying were significantly different, which corresponds to that mating behavior that I showed you through the animals. And this is about women who were married. If they were females with a female co-twin, they had significantly more children than a female with a male co-twin. And that corresponds to the finding I told you in rats about the greater fertility. So it looks like, and you know, we have much less information, but it looks like there is a fetal position effect for humans. So I hope that will convince you that maybe a little change in testosterone will matter for reproductive performance and various markers of fertility. So now I'm going to talk about this anogenital distance. So this is not a new measure. I was shocked to find a paper from 1912, first published data I could find on rodent AGD. And you can see this is the male anogenital distance, and this is the female. And you see they're very different. And in fact, if you work in a laboratory, you're used to holding up these little pups by the tail and looking at that, and I did it in Fred's lab. We could actually see, naked eye, which was a male and which was a female. And here are some numbers. But until this phthalate story came out, only one study actually had looked at it in humans, and mine was the first study to look at it in relation to a prenatal exposure. So the nice thing about AGD is that it's androgen-sensitive. It's used by the agencies that worry about reproductive toxicity. It's an endpoint. It's a reproductive tox endpoint. And we know now that shorter male AGD reflects an anti-androgen exposure. Now, I'm using this word very loosely. I'm not saying it goes through the androgen receptor. It may not, phthalates don't, but it interferes with androgen production. And the longer female AGD reflects an androgenic exposure, and that's pretty interesting. Girls with congenital adrenal hyperplasia who have excess androgen have a longer anogenital distance. I'm going to give you a male story, but there is a female story, but with much less information. So this is a very nice paper by Paul Fowler, 2011, in which he looked at abortuses No, I'm sorry, he did ultrasound. This is an ultrasound study where he measured, it was abortuses. There was another ultrasound study. Anyway, he looked at the AGD, and you can see as early as week 10, the males are longer than the females. And I think this is important, that this is starting to diverge and probably where we should be looking at exposures, critical exposures for this endpoint. So there's been a lot of work on AGD, and there's been a lot of work on phthalates. And here's a slide that puts them together and talks about a phenomena which has been called the phthalate syndrome. It's interesting, there's not too many environmental chemicals that have a syndrome named after them. This is pretty important. And so the mother is given, she can be given DEHP or dibutyl phthalate. They both have this effect, but in our hands, more strongly DEHP. Then you examine the pup after birth. And what you see is some external changes and some internal changes. So the external changes are reduced anogenital distance in the male, and these are all in the male, hypospadias, defect of the penis, cryptorchidism, undescended testicles, and then if you do surgery, you can look at malformations of the internal genital organs as well, reproductive organs as well. But we can't do that in our children. So together, these comprise the phthalate syndrome. This was published, darn, I'm sorry about that, in 2003 when I started, just about when John Brock asked me that question, and again by Paul Foster in 2005. And it got in the literature, it was pretty well accepted as a syndrome. And so I asked, because of John's question, is there a phthalate syndrome in humans? That's pretty important because it turns out that everybody in the United States is exposed to this stuff. So, you know, if it's causing the syndrome, it will be causing it in a lot of people. So to do this, I went back. Remember the study for future families? I had the urine. I had the opportunity to see what the children were exposed to because it turns out that these chemicals are non-persistent. They are measurable in urine. In fact, that's the best way to measure them. And we had the urine. So, and we had it stored well. And so all I had to do was get the babies. So that was tricky. I had to get money to do that. And I did succeed in getting that money and was able to bring back not a large number of babies because many of them were too old. Now think about what are you going to do? What are you going to do to look for a phthalate syndrome in humans? That's the question I posed myself. What should I look for? I talked to Fred. I talked to clinicians. I talked to pediatricians. And what I was trying to find was, okay, this causes a change in the genitals, maybe masculinizes the females. I don't want to say feminizes the males because that isn't what happens, but the males are less male typical in their genitals. And so what do you do? And so I actually had a lot of expert interviews and put together a protocol to examine the children. So that was the start of the measurement of AGD, and I have to tell you that now it is used worldwide. It's been published in thousands and thousands of children, and I may eventually become part of the standard of care because it's extremely, extremely useful. But this was a very exciting thing to be doing, and it all came out of the study for future families and the ability to do that here. So the study for future families, I told you about the first phase, that was the semen quality, but then the second phase was to measure prenatal phthalates in the urine and look at reproductive endpoints in the children. Now, think about what you have to do. You have to measure a distance in the genitals. You don't want to do that in 5-year-olds. They're not going to like it, and neither will their parents. Okay, so you've got to do it in young kids. Ideally, we think now it should be in a couple days after birth, actually, is maybe the easiest, or a couple of months. But at that point, the children were mostly too old. So we allowed them to come in up until 36 months. But most of them, their median was 12 months of age. And then we later went on to behavior, which I'm not going to talk about. So what we did, here's the schematic for the study. We got the mid-pregnancy urine sample. Now, mid-pregnancy, I showed you that Fowler slide. Mid-pregnancy is probably late. In fact, we know now it's late. There's some consistency of phthalate exposure over the pregnancy. That may help a little bit, but really you'd like to go early. So this study was limited in this. We had really too late. Our samples were late. We measured how completely the testicles were descended, giving a score for where they were placed. We got the penile width, we measured the anogenital distance, and the scrotal size. And a lot of subtle things like how much rugation, what is the color, and so on, things that you might look at, a pediatrician might look at in examining a 1-year-old child's genitals. And here's how we measured anogenital distance. And we're still doing it like this. This hasn't really changed, although we've gotten very, very good in our quality control and so forth. But, and we also, I should tell you, measure 2 things. This is the longer measure, so the center of the anus to the anterior insertion of the penis. There is a shorter measure right to here. See this point? The bottom of the scrotum? Okay, that is actually an easy landmark because the skin changes right there. That's a pretty easy landmark. And so you might think of this one as the shorter AGD and the longer AGD. And there's still a question about which of these is most useful. The longer one incorporates a lot of elements, right? The size of the scrotum, the size of the penis, the size of this anus-scrotal distance. So in some sense, it's more inclusive. It's also less precise because it's actually harder to find the top, to find that penile insertion is more difficult. So I'll just tell you that's, and we got pretty good at it. We could repeat it. In all of our studies, we've had every measure 3 times on every baby, the long and the short 3 times. And we also have a second examiner every 10th baby to get the between examiner variability. So when I asked the rodent people, how do they do this? I could not get a single protocol. So that's interesting. We do this, I think, much more consistently. We measured the 9 phthalate metabolites in this mid-pregnancy urine samples, and I'm going to focus, just for ease of talking, on one metabolite of DEHP. Now, grayed out here, these are also, this is a metabolite of DEHP, so is this. And so the bigger phthalate molecules have multiple metabolites. That's what we actually measure in the urine, urinary metabolites. And the smaller phthalates have, they just have the single metabolite. So what does this mean? So if you divide the total distribution of MEHP concentration in nanograms per milliliter in the urine, you see there, this 25th percentile, that's the point at which 25% of the mothers were less, and at 20, 25% of the mothers were bigger, had more. Okay, so, and then this is the middle. So we can just call this, for ease of speaking about this, low, medium, and high phthalate exposure. Okay, and now I'm going to show you a result. So it turns out that when we started out, we actually didn't know what was a short AGD. What does that mean for a boy to have short AGD? You know, how do you decide? And what should you control for in the analysis of these things? Well, it turned out age mattered, body size mattered, urinary concentration mattered. The center actually did matter despite all our efforts at standardizing. And in this first study, we actually made a mistake when we published this. We used a rodent method of standardizing for weight, which is dividing AGD by the weight, giving you an anogenital index, which actually doesn't control for weight. So for the rodents, it was fine. For us, actually, wasn't fine. So we had to publish another paper with a better control for weight, and that was weight for age. So for you who might worry about these kind of things, and probably not many here, we did control for all the necessary things. And then what we did was we just looked at the boys who were, you know, if you look at the difference between what we actually measured after adjustment to what we expected, what we actually measured to what we expected after adjustment, and how off were they? How much lower, shorter than expected were they, or longer than expected were they? And so they were, if they were in the lower 25th percent of the distribution in terms of being shorter than expected, we called them shorter AGD, okay? So that's where that comes from. It's very internal. There's no external standard. We were the first people doing this, okay? And now we know we actually didn't do too badly. But at that point, it was kind of, okay, just relative to the other boys in our population, that's all we can say. And by the way, we only had 105. Now we have a lot more. And so what this says is that if your urine contains MEHP concentrations in the upper 25% of the population, then you are 13 times more likely to have a boy with a shorter AGD. Pretty big number. And if you are in the middle, you have a medium level of DEHP, and this is for MEHP, but it's also true for the other metabolites, then you're about 5 times as likely to have a boy with a shorter AGD. These are big numbers for epidemiology, and this is highly statistically significant. And this paper made a big splash, I can tell you. It was chosen as the outstanding paper of the year by EHP, and it influenced regulation. So it played a big role in getting these phthalates removed from children's toys, even though we had made a lot of mistakes. Okay, so we also looked at these other endpoints. We looked at penile width, we looked at testicular descent, scrotal size. We had much fewer numbers, but these were also decreased in association with DEHP. So we had shown a phthalate syndrome in humans. What next? You do that, what do you retire? No, I think what you want to do is you want to do it again, and you want to do it better. And most people don't have that luxury, and I am so grateful to NIHS that they gave me the opportunity to do this again and do it better. And this is the Infant Development and the Environment Study, or TIDES, which is now ongoing. So now I'm going to tell you briefly about this study. So the number of women recruited was pretty similar to SFF, but the number of babies was much greater. That's because we built in the follow-up from the beginning. When we recruited women, we said, will you be in our study? Will you let us examine your children at birth? And then may we have permission to follow you? And so we were much more successful in that way. The centers were somewhat different. Minnesota stayed the same. I wasn't here anymore. So this is Rochester, New York, where I was. This is San Francisco, and this is Seattle, Washington. And we looked at what we had done wrong the first time and tried really hard to do it better the second time. And I think we really did. So we got women in the first trimester. Before, we had gotten them at a median 27 weeks of gestation. So this time we got them the first trimester. We got a questionnaire in each trimester and a urine sample in each trimester. We got serum. We did the exam right after birth, and then we also did a one-year exam. And now I'm happy to tell you that we're funded for follow-up. We just got this news a couple weeks ago that would confirm that we will be following these children for neurobehavior at 3 to 5 years. So this is what the study includes. Phthalate analysis of urine in the first trimester sample. We don't actually have a lot of money for analyzing all the samples, but we have the first trimester on everybody. We measured AGD in 758 children. So SFF had, with boys and girls together, about 220. So it's almost 4 times bigger. And we controlled for much better gestational age, age at exam, and this weight for length, the Z-score, which is a WHO measure, and maternal age, center-specific gravity, and the time of urine collection. And just to show you a little bit about how our population looked, so boys were a little bit heavier, you can't see it here, but they're a little bit longer. This is the shorter AGD measure. This is the longer AGD measure. And these are our results. So we found that these metabolites of DEHP, which are the ones measured by CDC, were related to both the shorter and the longer measure of AGD, and weakly to penile width. And they were very statistically significant. So when the bar goes way down, they're much shorter than they should be. And so this metabolite seemed to be the most strongly related, but there's not a lot of difference between them. This is the sum, the molar sum of the metabolites. So it's a good summary of all of these. You can just look at this one. So penile width is less sensitive, and the longer AGD is perhaps a little more androgen-sensitive, perhaps because it's incorporating all of those measures. And this was published actually just last month in Human Reproduction. So what's interesting and what made this really challenging is that between SFF and TIDES, the whole exposure to phthalates changed. And partly it changed because of our study. So, great, there was a reduction in exposure, 50% for these metabolites. So much lower levels. There was an increase in the substitutes for this, DINP, and in a companion study in Sweden, that also shortened AGD, so it's not clear how good a substitute it is. But we didn't see that. We have a different DINP than they do in Sweden. But these were not big differences. These are much smaller changes than we saw in our first study. And we were able to find statistical significance because we had been so careful and because we had a big sample size. So in all the ways that we improved this study, this made it possible to see this again. And what's really an interesting question is there a lowest level at which you would not see any changes? I don't know. Who cares about a short AGD? I mean, really, if you look at these boys, and this is what one of the press things said, Swan says, if you look at these boys, there's no problem, and you can't see any difference in their AGD. So everyone would go, who cares? Why are you even publishing this? Well, it turns out it does matter. Here are some ways it matters. Boys with, who have hypospadias and cryptorchidism have shorter AGD. This has been published now in a number of studies since our first study. That's a natural thing to look for. You have to do a case control study, and several of these have been done. In rodents, short AGD predicts low sperm count and problems with fertility. And so the question we asked is AGD related to sperm parameters and fertility in humans? Well, our children are too young. We can't study it. So we did something else. We said, well, okay, let's assume that AGD is going to stay with you throughout your life, and rodent studies suggest that, and we don't have that pinned down yet. We can't be sure. We need another 15 years for that. But we said, let's look at the AGD in young men. Let's look at their sperm parameters. Let's look at their fertility and see if they're related. And if they're related, that makes it plausible that these boys born with short AGD may have problems later on. And the answer was yes. So this study, which is the Rochester Young Men study, we put up flyers. Lots of men came to us. They said, for $75, we'll do anything. So we got a sperm sample, we got a urine, we got a blood, we got an exam, we got a questionnaire, we got their mother's questionnaire. And what we found was that both AGDAS and this is the shorter, this is the longer. You see here, the longer one is more strongly related to sperm concentration. This is not significant. This is significant. And in an adult male, you can imagine, particularly clinicians here, that the measurement, the location of the upper landmark is quite difficult, particularly if it's a fat person. I mean, it's just hard to find that upper landmark. So I think we know that this shorter AGD is measured with more precision, and this is the one that most studies in the world now are using. So if you want to use it here in Columbia, I would, and only had time for one, I would use the shorter one. And a colleague, Michael Eisenberg, in Harvard, Stanford now, looked at childless men and fathers, and what you see is that in childless men, the anogenital distance is very significantly shorter than fathers. And this, again, is the shorter AGD. And he also showed that the testosterone was related to AGD and the testicular volume was related to AGD. So it really does tell you something about the reproductive potential of those boys and those men. Now, I'm going to end by throwing a wrinkle in here just to wake you up. a very surprising finding, which we started to look at stress. And we don't have any sophisticated markers of stress. We don't have cortisol, we don't have biomarkers, but we happen to ask in both SFF and in TIDES the stressful life events question, because I had used it in the health department back there a long time ago. And the stressful life events is simply asking, since your pregnancy began or in the last 3 months, have you had a death in the family or serious illness? Have you had a divorce or serious marital problems? Have you moved? Have you changed jobs? There's 6 questions. They're easy, and they're really powerful. I'm not going to show you this, but in the RHYME study, the Rochester Young Men study, it was very predictive of sperm count. Young men who had a lot of stressful life events had 33% lower sperm concentration. And in this study, we found a lot of associations with this question in pregnancy. And in rodents, maternal stress induced a female-like anogenital distance, delayed testicular descent, and no change in females. And I haven't talked about behavior, but we also have some findings on behavior consistent with prenatal stress feminizing male behavior. So it looks like in males, stress is anti-androgenic. We're not sure. It's definitely playing a role. So we wanted to look at that. And actually, we have a grant now on that. So I want to show you what this means for TIDES AGD. So I showed you a picture of the results of AGD and the DHP metabolites. Now here it is in a summary form. And just let's again look at this MEHHP. And what you see that overall, this is the longer, this is the shorter, and this estimate looks a little bigger, but they're pretty much the same. You can see that there's a significant association that higher MEHHP means a shorter AGD. But it turns out that if you limit this analysis to boys whose mothers reported 1 or more stressful life events, that's not a lot, but these are big events. So we call them highly stressed, but you know. There's nothing. Look at this. Nothing. All gone. It's really remarkable. And if you look at, come on, the mothers who reported no stressful life events, you see a stronger association. So all of this association between phthalates and AGD is in mothers who were not stressed. I don't have the answer to this. I'm just throwing it out as a provocative, something to provoke you and think about. This is modification by stress of phthalate AGD association. And finally, I just want to summarize. We also looked at SFF. So in SFF, interestingly, when the mother reports more stress, the girls are somewhat masculinized, suggesting that in girls, stress is androgenic. And Fred has seen that in animals. In boys, there's a suggestion of a less masculine, now in SFF. So we saw in SFF that stress significantly modified male AGD phthalate association, and we also saw it in TIDES. So we have now an R01. My mentee is PI now, and she's got this funded, and we're going to be looking at this in much more detail. I think it's a fascinating story. So I want to single out Emily Barrett. That is the person who got the new grant on stress, and these are the people in our studies. Irma Drobnitz. Is Irma here? No? And funding from NIHS and US EPA. And this is our TIDES family, and I'll take questions. Thank you.
All right, I'll start. Fred, you've got a microphone, so you might as well start.
So I just wanted to mention that in the animal studies, you're going to be looking at behavior, and we followed animals with large or small anogenital distance, males, throughout their lives. And one of the things you see is all the things you'd expect if you drop testosterone in the low anogenital distance animals. They're less aggressive. Their sexual behaviors are different. Their activity levels are different. Their ability to metabolize drugs and chemicals are different. Their livers are different. And their brain structure is different. So the areas of the brain that control reproductive behaviors are different as a result of that difference in testosterone. None of this is surprising. If you don't see behavioral effects, I will be astonished.
Oh, we have seen behavioral effects. So in SFF, we have, no, I got this. In SFF, we followed the children twice, and we found the same thing you said, that the boys more exposed to phthalates played in a less male-typical manner. We used a preschool activities inventory, and then we looked at their cognition and behavior at ages 6 to 10, and we found effects that perhaps somewhat different than you'd expect. We found that the boys were had more behavior problems and poorer attention. In TIDES, we're looking at a bigger battery. We're actually examining the children, so we'll have more information.
Two questions. First, do you think there's anything resembling a ceiling effect where in the stress you've already maximally altered things such that the phthalates don't have any additive properties in that situation in the combined effect, maybe that being the mechanism of the loss of interaction? And the second question is a more open-ended, love for you to comment on the changes in blowback that you've received politically over the past couple decades for discussing findings such as this.
Okay, well, in terms of, I mean, what I think is going on in the stress modification of the phthalate AGD association is that, and this is pretty simplistic, so it might be, could be totally wrong, just guess. But I think that because we see in girls that stress is androgenic, it may be that it's also true in boys, although we haven't measured that. We haven't seen that. We've seen hints of it. But if stress is androgenic, or this kind of stress, oversimplified, if stress is androgenic, it may counter the anti-androgenic effect of the phthalates, so kind of balancing out these two things. And so the women who are chilled out, who don't have this androgenic influence, may be the ones in whom you see the effect of the phthalates. So that's my guess. But now, as far as the blowback, are you talking about from industry? From,
yes.
Yes, okay. Oh my gosh.
I was talking to Fred about this a little earlier.
I have to show you all the best thing. I didn't show you the best thing. Okay, this is blowback. I'm so glad you asked. So when Fred and I were on the committee for endocrine disrupting hormonally active chemicals, they made us call it, we got attacked. And we got attacked as endocrine disruptor crybabies. And we made these T-shirts. Okay, endocrine disruptor crybabies. Well, there were 4 of us, and we actually, wore them. So Fred saved his, and I want to share this with you. We did get a lot of blowback, and but that's fine. I mean, people make chemicals. That's their business. We try to study their effects. That's our business, and that's what.
Has it changed over time, though, or has it just been pretty continuous?
What?
Has it changed over the last couple decades, or has it been continuous?
I think the concept of endocrine disruption is now accepted. It's accepted by many major medical societies, endocrine societies. So we don't get the same. We're not called truth disruptors. That was another thing we were called anymore. But it's very controversial. It's very controversial. And I also, yeah. So we just have to keep on doing it and do the best job we can and deal with it.
Yeah, my question was sort of answered by Fred's comment, but with the graph where you showed that the correlation with the sperm count and the ADG, AGD. And then you showed fatherless or childless. And so my question about that was kind of sexual preference, right? Was that asked in the questionnaire? Because if there is some indication that this will be less masculinized or more feminized, then that may kind of account for some of these effects that you see here.
These were all people seen at a fertility clinic, so maybe that's not the best place to look for that, but it's a very good question. I mean, I think we did see that boys exposed to phthalates, and also boys with shorter anogenital distance, were less masculine in a lot of ways, including play. And they're too young. We don't know what their sexual preferences are, but I think that's that's something that should be studied. I just don't know. And it's hard because I think you can get an AGD measurement on a child up to age 2, 1.5, and then you can do it again when they're maybe 17.
So how old were the young men that you studied?
Oh, and the Rhymes young men, they were college students. Yeah. And they volunteered and they knew what they were getting in for. So they, but to have a kid come into a pediatric office and start measuring their genitals, well, would you do that? Oh, yeah, but they're cases, right? I mean, they're not routine, right?
They should get you measure babies.
Yeah. Babies, yeah. Yeah. But 5-year-olds?
You should get a good genital exam now.
Yeah, you think you could do it? I mean, I would love to do it, but the feedback I got from study sections and from our IRB is that this is too invasive for a child over, say, 2 years of age. Yeah, please.
The differences in play and all
Talk-
of
I
these,
still can't
the
hear.
differences in play and all the things that have been looked at in children with autism, Simon Baron-Cohen's work and Scoos's work. Has this been done in autism or Turner syndrome?
Can you do it?
Pardon?
Could you do it?
Oh, yeah.
I mean, we should talk, because I think I would love to have this in, or Irva. Could you do it? Could you do it?
Well, we have a cohort. Unfortunately, some of them are past 3 to 6 months.
But Judy's saying it doesn't matter. I think it would be fantastic. I don't even know what I would predict, honestly.
So, yeah,
What do you think?
that was my question
Yeah.
to you is, do you think phthalates are going to, would you expect that you would see reduced risk for autism? I mean, Baron-Cohen says this is extreme male brain and so forth. So, but there are 3 studies now that show,
I mean, if we go by, the Baron-Cohen's theory that it's the extreme male brain, it seems like you'd be predicting the other. But there actually are now 3 studies. 2 used the social responsiveness scale, so it wasn't a diagnosis of autism, but it was looking at symptoms of autism in more general populations, and those studies actually do show phthalates associated with more deficits in social interactions, more deficits,
Yes,
poor social
that's
things.
what we see also. That's what I was saying.
And then the third study was the one in is it Sweden? I think it's Sweden, which was the something like, I forgot what the name of it is. It was a study of asthma and home exposures.
Selma,
Is that what, okay, you're right. And they saw that if there were more rooms in the house with vinyl flooring, or maybe it was more likely to have the I think it was the parents' bedroom or something.
Yep.
Anyway, the vinyl flooring was associated with a greater risk of autism, and vinyl flooring, emits high levels of phthalates into the home. So, so there, there's a little bit of data. We have some data that's in review right now. So, but it seems like the androgenicity aspect seems like but Baron-Cohen could be totally wrong. In fact, I wouldn't have that much trouble believing that.
Yeah.
So.
Yeah. I don't have a prediction, but just to say that the phthalate story and the AGD story are not the same. There are lots of things that affect AGD. So, I mean, I would be interested in seeing, because AGD is, no question, a measure of the androgen environment in utero and a predictor of certain later development. So, aside from phthalates, it would be worth looking at.
So one question I have for you is, what is the relationship between the amount of androgens and the actual androgenic activity of those androgens? And
we have a study ongoing now on the mother's steroids, and we'll have some information about that. But that doesn't tell you what the level of androgens are in the fetal testes, which is really where you want to go. So I don't know any way to answer that question.
Shanna?
Yeah,
Sorry, I just want to call your attention to the question over there.
Michael?
I think I'm first going to stand up for Columbia Men, or stand erect. And what my recollection of that study was that it was done once.
Yes.
It was done between 4 or 5 different places with different individuals assessing sperm quality, and that is quite critical when assessing sperm quality. And so there's always been a certain amount of skepticism in my mind as to whether that really did reflect differences. So that was one question. And the last one was when you got this overwhelming effect relating to the phthalates and the anogenital distance, it was something like 14-fold or something. You only had just over 100 people in the study. And so how many of them were actually in that group?
That was the 25%. So there were 105 boys, so it was like, 25% of 105. But don't forget, we replicated it then with 363 boys.
Because the effects were nowhere near as large.
No, but they were consistent with the same effect at a lower dose level. They're totally consistent, actually. But let me just say about the semen quality. You're right. This quality control for semen studies is a big issue. We did as well as can be done. We had monthly standardized samples circulated among our centers. We had a control center in UC Davis that sent out blind samples, and when the examiners measured the sperm, sent the results back to Davis. Davis compared them among, and we did that all throughout the study. If they were drifting, then they were retrained. We had the same instruments at each site. We had a central training for all examiners. So, and those results are all published. That quality control results are very, very good. And our data has been used in the WHO manual. It's considered very reliable data. So, what it means, that's a whole different question. But all I can say is at that time, I'm convinced, not now necessarily, but at that time, sperm count was lower in that group of Missouri men. That's all I can say.